Drug development is very expensive. The calculated cost is around $1.5 billion per new drug. It takes around 10 to 15 years. The major reason for drug development to be so expensive is because of what we call clinical trial failure. We have a 90% failure rate. IVAL proposes a new paradigm for drug development, which is, at the beginning, evaluate drug properties with human-based in vitro system to choose drug candidates that are most efficacious and the safest for further development. In vivo testing with the wrong animal species, even though you may use 2, 3, or 4 animal species, if they don't have the right metabolic capacity like human, it will not give you the right answer. By cutting down the failure rate in the clinical trial, you can cut down the cost of drug development as well as the time of drug development. Our idea is that hepatocytes from the human liver or human hepatocytes can be used to understand drug properties in the human liver.
Dr. Lee, some 25 years ago, invented the process to cryopreserve hepatocyte tissue. We freeze them, we make a consistent single lot from a batch of liver, and then we can make that available for ADME testing and pharmacology testing and tox testing in the industry. So what this does is it provides a complete system to get human data and predict what it will do when it's in vivo in the clinic.
With hepatocytes, when they are thawed from cryopreservation, that would have over 90% viability, that would have a near 100% plating efficiency, and would stay in culture, retaining morphology and properties for over 9 days, what we call 999 elite human hepatocytes. So now we have the highest quality cryopreserved human hepatocytes.
IVAL and Dr. Lee's reputation is worldwide. Our clients range from the largest pharmaceutical company to the smallest biotechnology laboratory. Our customers cover about 34 states across the US, approximately 25 countries in Europe, Canada, Japan, China, India, and South Korea.
Cell culturing is a skill, almost like an art form. There's a series, it's kind of like cooking. There's a series of ingredients, but not everyone makes the same food at the end of the day. What we do here is we provide with the product the plated cells. And then for different systems, we deliver those plated cells by hand. So it's less than half an hour typically for them to go from the incubator here in a controlled delivery to the lab where they're going to be used. And that can be anywhere from a couple of hours old to a couple of days old. Depends on how mature the cells have to be for the process that lab is doing. It's a tremendous resource. It takes the weight off of having to keep that talent in-house, which the current environment has made difficult.
We have perfected cryopreservation procedures for human hepatocytes, and we have extended those procedures now towards human enterocytes. The intestine is an organ that is very important in terms of the total metabolic fate of the drug. IVAL is the only company with cryopreserved human enterocytes, cryopreserved human intestinal mucosa, 2 systems that retain intestinal drug metabolic capacity. Combining intestinal metabolism and liver metabolism, we will have the entire picture of how a drug is metabolized in the human body.
One of the exciting features of IVAL are the workshops, training, and symposia we hold with top presenters in their field. One of our workshops is hepatocyte use training. Then we have our yearly drug-drug interaction conference that covers novel technologies, enzyme induction and inhibition, regulatory, and drug-drug interactions.
A recent development in the medical field is the application of gene therapy to combat diseases. And we have applied our hepatocyte to understand the efficacy and duration of efficacy of RNA therapeutics. And that is something that's very exciting. This approach of placing importance in vitro system to understand human-specific drug properties is something that is uptaken, accepted now by most scientists in drug development. That is something that I'm very proud of. Our goal is to accurately predict how a drug would act in a human body for both efficacy and safety.